my work is trying to understand what are those electrical events that underlie complex cognitive functions such as memory and learning, and try to understand what goes wrong in that electrical activity in case those cognitive functions are for some reason impaired, like in the case of dementias and specifically in the case of Alzheimer's disease. It is key that the animal has a nice life, basically, right? If you're studying a disease or a physiological condition, you want to standardize your conditions as much as possible. The environment where the animal is will somehow affect the end results. So you want to have control over that environment, especially over the possible stress and pain, because those factors might contaminate, might introduce a bias into your results. So basically what you can see on the screen is a section of brain. This is called acute section of brain because it's alive and it's been excised by the brain of an animal. The animal has been humanely sacrificed with no pain nor stress, and the brain rapidly removed and sectioned by keeping it immersed into an artificial cerebrospinal fluid at 4 solution at 4 degrees. The brain slice is positioned into a perfusion chamber. So the perfusion chamber is integrated with an optical system, a microscope, that allows me to actually visualize single cell bodies, single neuronal bodies, and then what I'm going to try is to use this micromanipulator system to approach the cell with a glass micropipette filled with a solution that resembles the composition of the innards of the cell, and I will try to patch the cell, which means that I will try to suck the plasma membrane of the cell onto my glass micropipette, break the patch of membrane that is between the cell and the pipette, and so record anything that is coming from inside the cell in terms of electrical activity. So these actually are all cell bodies embedded within the matrix of the brain. And the advantage is that with an excised slice, I can actually visualize and I can record the activity of these cells in a almost perfectly native environment, at least much more physiological than growing these cells into a petri dish. So what I will be doing now is to fill this pipette with some solution. This is an artificial solution which wants to resemble some of the features of the internal composition of neurons, especially in terms of the composition of the electrical ions, okay, like sodium, chloride, and potassium. The concentration of these ions are key for driving the electrical properties of a cell. So now what I do is to place this pipette onto this head stage. This head stage is connected to an amplifier, and so the first task is to find the tip of the electrode. So that is the pipette, as you can see. So this is the slice, right? I just want to position my pipette just above it. So see, I identified a very nice cell body here, and so what I will try to do is to approach it with my pipette and to form a seal. Yeah, so see, now the seal is formed, and all these little blips that you see, those are neuronal activity. Now what I can do is to show you the spontaneous firing of the cell. Okay? So this is really how the cell behaves in real life. And these spikes are the ways cells communicate with each other. What I can do is to measure the different, for example, waveform properties of the single action potential between an Alzheimer's mouse and a normal one, because the shape of this single event decides how the neurons talk to each other, and the way the neurons talk to each other decide how a cognitive function works or doesn't work. There's been a lot of advancement in Alzheimer's research lately, especially on the genetics of Alzheimer's disease. Now, we don't really know what is the mechanism that goes from those genes to the final clinical outcome of the disease, to the dementia itself. What I'm trying to do here is try to record the electrical activity of the brain of certain mice that have a specific gene related to dementia.